Molecular biology is a branch of biology that involves the study of biological molecules, including DNA, RNA, and proteins. It has revolutionized our understanding of the genetic basis of living organisms, and it has led to the development of numerous techniques that have transformed the field of genetics. In this short video, we will review in this lecture some of the key concepts and techniques of molecular biology, with a particular focus on genomics. The concept of genomics is the study of the entire genome of an organism, which includes its complete set of genes, as well as all of the non-coding DNA sequences. Genomics has transformed the way we study genetics, as it allows us to analyze the entire genetic code of an organism, rather than just individual genes. This has led to a deeper understanding of the genetic basis of complex traits and diseases. The structure of a protein-coding gene. A protein-coding gene is a segment of DNA that contains the instructions for making a protein. The structure of a protein-coding gene includes a promoter region, which is responsible for initiating transcription, and a coding region, which contains the instructions for making the protein. The coding region is made up of exons, which are the parts of the gene that code for the protein, and introns, which are the non-coding regions that separate the exons. The genomic organization of prokaryotes and prokaryotes, such as bacteria, have a simple genomic organization, with a single circular chromosome that contains all of the genetic information. Eukaryotes, on the other hand, have a more complex genomic organization, with multiple linear chromosomes that are contained within the nucleus. Eukaryotic genomes also contain a variety of non-coding DNA sequences, such as repetitive sequences and transposable elements. The types of sequences within the eukaryotic genome, coding and non-coding sequences. Eukaryotic genomes contain two main types of sequences, coding sequences and non-coding sequences. Coding sequences are the parts of the genome that contain the instructions for making proteins, while non-coding sequences are the parts of the genome that do not code for proteins. Non-coding sequences can include regulatory regions, such as promoters and enhancers, as well as repetitive sequences and transposable elements. The basic facts about the human genome. The human genome is the complete set of genetic instructions for making a human being. It is made up of approximately 3 billion base pairs of DNA, which are organized into 23 pairs of chromosomes. The human genome contains approximately 20,000 protein-coding genes, as well as a variety of non-coding DNA sequences. The relationship between genome and proteome. The proteome is the complete set of proteins that are produced by an organism. The genome provides the instructions for making the proteome, as the DNA sequence of a gene determines the amino acid sequence of the protein that it codes for. However, the relationship between the genome and proteome is complex, as there are many factors that can influence protein expression and function. The potential applications of genome sequence data to medicine. Genome sequence data has the potential to revolutionize medicine, as it can provide insights into the genetic basis of complex diseases and disorders. By identifying genetic variants that are associated with disease, we can develop targeted therapies and personalized medicine approaches. Genome sequencing can also be used to identify individuals who are at risk of developing certain diseases, allowing for early intervention and prevention. The importance of comparative genomics in revealing conserved elements that are likely to have interesting functions. Comparative genomics involves the comparison of genome sequences from different organisms, with the goal of identifying conserved elements that are likely to have interesting functions. By comparing the genomes of different species, we can identify regions of the genome that are conserved across evolutionary time, indicating that they are likely